Very good. Thanks so much, Karen. I'm excited to be here. Thank you so much, University Lab Partners, for giving me this opportunity. So today I'm going to talk a little bit about the pitfalls, strategies, and opportunities in vaccine manufacturing. And I've entitled it Walking Softly Through the Minefield, since there are a lot of pitfalls to avoid along this path towards opportunities. Let me first, we're at the beginning of the talk, but let me first just ask a question. I wanted to get an idea just for the audience, what its makeup is. Samuel, if you could put up our first poll, I'd like to have an idea we're primarily made up of bio-entrepreneurs, people with just general interest in the field, active vaccine researchers / scientists, or even anti-vaccine activists. I know that's become very big lately. So if you could do that and submit, I'd be very interested. This will kind of help me to fine tune the talk as best I can on the fly and be able to maybe address some of your concerns or just tailor the talk in response to the actual makeup of our audience. And Samuel, I'm going to ask one more question, one more poll of the audience before we get started also. I wanted to get an idea for how long people think is a reasonable time frame to develop a vaccine, to take it from its very inception, and actually take it into the clinic. If you can put that slide up, Samuel, that'd be great. Do you think it's reasonable for 6 months? Do you think it's a year? Do you think it's 2 years? Do you think it's 5 years? Or even 10 years or more? There are a lot of different opinions out there. A lot of folks, I have a good friend, he thinks 18 months is absolutely absurd, that it takes, should be much shorter than that. So I'd be very interested to know what the audience thinks. Interesting. So most folks have about 2 years. So let's see what's sort of like in the popular media, what public figures, what politicians tend to think on the subject. Before I get too involved, just want to read a disclaimer. The work included in this webinar is based on peer-reviewed publications, SEC filings, interviews, corporate press releases, news articles, and other forms of publicly available information. Any investment decisions should not be based solely on what is presented during the course of this webinar. This webinar may contain forward-looking statements that involve known and unknown risks and uncertainties, and none of the statements made are on behalf of a publicly traded or private entity. The views expressed in this webinar do not necessarily reflect the views of ScienceDocs or its management. The content of this webinar is the exclusive property of ScienceDocs and is not to be shared or disseminated without the express written consent of ScienceDocs. So now that I've got that basic piece of housework underway, let's have a look. So you're hearing a lot of different things in the media and a lot of different things from high up. And these are very, very different perspectives. On the left, that man needs no introduction. That's our president. And look what he said. I've heard very quick numbers, a matter of months, and I've heard pretty much a year be an outside number. So I think that's not bad at all, not a bad range. But if you're talking about 3 to 4 months in a couple of cases and a year in other cases, wouldn't you say to a doctor, would that be about right? So as of June 5th, 2020, there are 2 million corona vaccine doses that are quote unquote ready to go. One scientist figure whether it's safe and effective. Then we've got Dr. Fauci, all right? So he's saying if COVID is like other coronaviruses, it isn't likely to be a long duration of immunity. He's talking about the actual infection itself. And he says the US should have a couple of 100 million doses of COVID-19 vaccine by the start of 2021. And he said that on June 3rd, 2020. He said that as a start of 2021, and they said that's a slight typo. As on June 3rd, 2020, he said that, and then there's a link, so you can go see that yourself. The reality is or a different opinion, I should say. This comes from Sanofi Pasteur during my time as a contractor there, in their seminar on understanding our science, is that most vaccines never make it to the market. Some make it, but they don't reach their original product goals. A few make it, they perform as designed, and this can take 10 years or more to complete. So that's quite a contrast to what the president and what Dr. Fauci are saying. So let's try to get a handle on these different timelines and understand why it might take so long and why thinking something like you're gonna have a couple million doses just sort of quote unquote ready to go is really not realistic at all. So first, let me talk about the challenges to vaccine development, why you can have such a long timeline. And the path to developing a successful vaccine, it's a heavy load to lift. Got this little picture from one of the world's strongest men competitions, in case anybody's curious. So what's going on here? What are the obstacles that we have to surpass? Most vaccines require boosters, so that means you can't just get a single shot, one and done. That just doesn't really exist. There are missed opportunities to vaccinate. Sometimes certain people oppose vaccinations for various reasons. Other times they don't get it early enough. Mass production, huge. I mean, you can't get something to the patients if you can't make it. It's that simple. Everything has to be made, and it has to be made at large scale, and it has to be made safely. It has to be made reproducibly, and it has to be made under GMP conventions. Not so easy to do. It's expensive. Some folks have completely left the vaccine game because it is expensive and the rewards aren't so great. Another challenge: there's no centralized database of vaccination status. So it's very hard to tell who's been vaccinated, who's not, how to kind of keep track and manage that. I've got a question on this subject for folks a little bit later in the talk. Possibly adverse reactions. Those exist. But fortunately, they're usually on the rare side. And then less than 100% efficacy. So even if you get a vaccine, you're not 100% protected. You can't just go, "Whew, I'm safe. I'm safe for the season." Let's consider flu. It's a well vetted vaccine, been around for a long time, but you're only batting 50 to 60% during. It really depends on the season, but it's nowhere as close to 100%. It works. It's better than nothing. If you do get the flu and you had the vaccine, you're way less likely to suffer a serious complication. So in that respect, it's good, but it's certainly not at 100%. And then, the vaccine, this is all borne out in my final bullet point here. The vaccine's licensed for use in the United States. So I would encourage all the attendees, I believe this is a live link. It's not just cut it out, put it in your browser. Go and have a look. For me, this list was shockingly short. I thought it would be quite a bit more products that are licensed for use in the United States, but it's really very, very few. And I should mention that today's talk, I'm going to concentrate on vaccines directed towards viruses. I'm going to leave the ones towards bacterial infections alone, since the talk will just get too broad. Just sort of as a little qualifier, but it really is striking to see just how few of these we have that are approved. And I should add, probably there's another obstacle there, is we live today in a very litigious society. I was, for something like measles, right, a very well vetted vaccine, using an attenuated virus, to actually prove that it doesn't cause any sort of disease. It would be, if somebody actually started out de novo trying to work with the measles vaccine, because of the implications, because of legal liabilities, probably it wouldn't even be attempted. So you've got some real powerful forces that are opposing vaccine development that you have to be aware of. That being said, it's certainly a worthwhile endeavor, because when it works, it's a very, very powerful tool to fight disease. So, getting back to our time frame, understanding why it takes so long. If you choose to go this route, you have to understand there's 6 phases of vaccine development. And let me just tell you a little bit how it's sliced. Right, you start out at your very beginning. That's the preclinical, before you go to clinic. You're going to work with animals, in vitro systems. You're going to look at toxicity, immunogenicity, the potential maybe to combine different antigens or viruses for a combination vaccine, stability data and potency, correlates of protection, right? You'll look and see like antibody titers in animals, if they actually correlate to protection when exposed or challenged by the virus. You'll begin developing your manufacturing strategy, all right? And then that will all culminate in the filing of an investigational new drug filing, right? An IND. All right, so then once you're on your IND, you're going to make the jump to phase 1, right? And that's typically quite small, right? If you're going to, for one of these studies, you'll have 10 to 40 human volunteers. Again, we're talking about a vaccine trial. It'll go weeks to months. You're primarily looking at safety, all right? And you're also you're going to examine dose size and the route of administration. You're going to look a little bit at mode of action and immune response, and also you'll get to understand the characterization of your vaccine a little better. If phase 1 proceeded properly, everything's safe, then you're able to go to phase 2. And that's a little bit bigger in scale, right? That increases in scale and increases in complexity. Now you go from 10 to 40 human volunteers, 10 to 40 healthy human volunteers usually, to 50 to 500 volunteers. This will range from months to years. And you'll look at rates and severity of common adverse events. You look at the immune response of target groups. If that goes well, now you're into phase 3, right? That can go to 2 to 5 years. So now you've got several thousand, right? Anywhere from 20,000 to 50,000 volunteers. I mean, it's pretty, now you're getting a really large scale. Placebo-controlled, you look at safety, efficacy, establishment of final manufacturing procedures. Typically, you're gonna have validation lots or consistency lots. You're gonna make 3 lots around your manufacturing scale. You make them consecutively, and you're going to show that it's a reproducible process, that all of your critical quality attributes, your acceptance criteria, are consistently met. And if that goes well, then you get to a BLA filing, and that might take 1 to 2 years in that process, right? And so I made a separate number 5 for the BLA approval, the back and forth, the filing, and then even then, once you get approval, FDA approval, you're still not done, because now you have to have post-market surveillance. You're monitoring after licensure, safety, efficacy, new indications. So it's sort of a never-ending process. So you can see just how long this takes, and that's just to get you to your first vaccine. You'll see in my final slide, I talk a little bit how the first vaccine's typically not the best. It's an iterative process. So even once you get something that's approved and you're monitoring it at the post-market surveillance level, you still may improve upon it in years. So it can take years, if not decades. So it's really getting people's hopes up, raising hopes unnecessarily, giving people's false expectations to think that you can just have something that's any good. And most importantly, is safe in just a matter of months. All right, so I mentioned the previous thing about combination vaccines. Figured I'd like to talk to them a little bit. We've got a bunch of bio-entrepreneurs in the audience, so this is sort of a useful tool, a useful thing to develop. You sort of get more bang for your buck if you can do it. What's the most typical combination vaccine? Probably the MMR vaccine, the measles, mumps, rubella, typically that young kids get. That's probably the best example I can think of off the top of my head. So, like I said, you're getting several for the price of one, right? You're preventing multiple diseases, or you prevent one disease caused by different strains or serotypes of the same organism. You offer the benefit, right? Fewer injections, but one of the challenges, now stability, which I'll talk about later in the talk as well, is it becomes a little bit more complex. So each component in your mixture has got to be stable, and each component of the mixture you've got to understand its potency. So you're going to have understand the potency of each virus or each antigen in and of itself, and then in tandem with its other mates in this combinatorial regime. So again, you're going to still have to do you have to do your validation lots, the component lots, right? They've got to represent 3, preferably consecutive production lots. The adverse events, right, of your combination vaccine, they got to be less than or equal to the adverse events seen in component of each individual component. The immune response, protective levels, right? It's got to be, again, in tandem with its other components in the mixture. They have to be at least at the same level as they were individually. So each bulk component's got to be tested for identity, sterility, and purity. So it's a little more complicated, but if it works, you have a very powerful tool in your arsenal. And I should say, you can read more about this if you're so inclined, right? There's a guidance, FDA guidance, for the guidance for industry for the evaluation of combination vaccines for preventable diseases, production, testing, and clinical studies. Okay. So that takes us to our next slide. So you got to understand there are multiple types of viral vaccines. And this is a breakdown. This is just sort of how I see the field. I've tried to make it as straightforward as possible. And I'll talk a little bit about the different classifications, the different categories, and an example from each class. So you've got the live attenuated ones. So this is a virus, right? It's live. It's typically attenuated. So unlike the wild type virus, it doesn't really cause a disease at all. If it does, it's a very, very mild disease. What do we got there, right? You got YF-Vax for yellow fever. You got Varivax. Those are two members of this class. Another one is like you've got a vaccine, right? You've got a virus, but now it's either been killed, right? The virus is intact, but it's just been inactivated. And typically you might use a reagent like formaldehyde to kill the virus. It's no longer infectious. If you expose it to cells or an animal or a human, it won't cause an infection. It's dead, but it is antigenic. So it's killed and activated. Sometimes when you say subvirion, it's actually split. So not only have you killed the virus, you treat it with some sort of detergent, and it breaks up into little pieces. The flu vaccine from Sanofi Pasteur, that's a wonderful example of a killed vaccine that's also split, right? And that's Fluzone, right? And then Ipol's is a vaccine for polio. Then another one's called another classification will be the recombinant vaccine. So now you'll just take, with the magic of genetic engineering, you can get the cDNA of a component of the virus, express it in a recombinant system, and then use that artificially expressed protein, that fragment of a virus, and immunize people. They'll mount an immune response against that fragment, and that will be enough to actually fight against exposure to the intact live virus. What are good examples? Shingrix, vaccine against shingles, Recombavax HB. Actually, that's not really a viral vaccine, so I cheated a little bit, but you get the picture. Then it gets a little bit sort of more artificial to kind of go down the spectrum, right? We go from live to now virus-like particles. So these aren't really viruses at all, but what they are, it's a tremendously cool technique. Where you take the different structural proteins of a virus, and in a recombinant system, you're able to express those, have them pieced together. So it's almost like it can be like a hollow shell of the virus. So it's not infectious, right? You don't have to worry about anybody getting infected, just not a problem at all. And there's some commonly available vaccines, Gardasil, Cervarix, right? Those are all virus-like particles, FDA approved. I put up there another one in case anybody's curious, Hecolin, that's not approved in the United States. Put a little footnote there to that effect, but it is approved for use in China and for the prevention of hepatitis E. Then you have genetically engineered vaccines. So this is where you'd have a virus, right, an unrelated virus, and you take the viral protein from your virus of interest, and you'd express it in the context of a different virus. So a virus that's really not terribly infectious, that really won't cause a lot of harm to a person, but now it harbors just a protein from the virus that you're trying to vaccinate against. So that's a different approach, also very interesting. Now, as we go down on the spectrum here, we're getting to more and more chemical approaches. DNA, RNA, to tell you the truth, there's a lot in the news about this, but there are none that are currently licensed. DNA takes a bunch of different forms. Most typically, they're plasmid vaccinations, right? Where you've got a circular DNA containing your gene of interest. RNA also, that's quite a bit in the news, where you inject the RNA that gets translated into protein, and hopefully you mount an immune response against that. And then finally, at the list, down at the bottom list is sort of our most synthetic, and that's the peptide, right? It's a short stretch of protein, chemically synthesized. Field's been around for a long time, but none are currently licensed. So, of the list here, I'd have to tell you that I think a peptide-based vaccine probably is the least likely to come to fruition. And I base that on, there's 30 years of folks out there doing research on this. And it's really has yet to come to fruition. I'll talk a little bit more about it, but the only real utility I could see for a peptide vaccine is not so much mounting an immune response, but maybe if it acts as a decoy and it somehow prevents a virus from docking or it disrupts a viral replicative protein, that might be a use for it during the course of infection, but I don't think as a prophylactic agent. I could be wrong, and I certainly hope that I am, because if I am wrong, that means people in the end will benefit. But history sort of is weighing very heavily against this molecular candidate. Okay, again, now that I've sort of got going a little bit on peptides, I don't want to spend too much time on it. I'll give you a brief overview since it's out there. People are working on these things. What actually is going on? Well, they're typically very poor immunogenicity, right? Usually you would take a defined region that's associated with protection of the viral protein. When you deliver it, right, you've got to worry now the peptide's highly susceptible to proteolytic cleavage. So you've got to get something that's stable. You probably have to have some sort of chemical group on it. It's going to have to be immunized in the context of an adjuvant, or else you'll never mount an immune response to it. A lot of times you're trying to, humoral immunities means the level of the B cell. You're trying to get your body to generate antibodies against a foreign agent. Well, they really aren't just short stretches of proteins. These are proteins with an intricate fold, a three-dimensional conformation. Very hard to duplicate that with a peptide. Problem is now, with regard to cell-based immunity, peptides have a tendency to actually be tolerizing, right? If they're not uploaded properly in antigen-presenting cells. That is, if they don't go through the antigen presentation pathway, they can dock, outside of the cell to the MHC and actually act as a induced tolerance, which is really the opposite of what you're trying to do. Right. So, okay. So yeah. So then it's then like I said, just sort of weak immune responses in general. So, like I said, you've got to have either your adjuvant, some other sort of protein like that comes with it, like the KLH or complete Freud adjuvant. You see those a lot in mice or murine systems, but it's really not something that's likely to ever get FDA approval and be used in the context of a trial on humans, or that can be made on a large scale and done on people. So enough said about peptide vaccines. We're kind of going the opposite way, which I initially presented the list, but now you got the DNA RNA vaccines, right? So, and I kind of put these guys together, for both of them, you can see the use of vaccines, it's attractive from the standpoint of manipulation, manufacture, and standardization, right? You can change, that is, you've got a genetic code, right? You can change a single base pair, right? Single base. And that could change like an amino acid sequence. So they're tunable, so to speak, right? Non-infectious. You don't have to worry about anybody getting infected. You don't have to worry about like a live vaccine that might revert into an infectious disease and get somebody sick. They've been, when I put these facts up here, these are all things from peer-reviewed publications. Both have been documented to stimulate innate immune responses, right? The innate immune system is not talked about as much as the adaptive immune system, but it's certainly equally important. You can also, both of the RNA and DNA modalities, they can induce T and B cell immune responses. That's all been documented, right? There are some concerns, right? If they're taken up by an antigen presenting cell, right? There's something called original antigenic shift, which basically is just, there are different terms for this. You also talk about antibody dependent enhancement, but in a nutshell, it's just sort of a lack of diversification of an immune response where you kind of get locked in for one response for one class of viruses. Well, then what happens if a different serotype, a different variant of the virus arises, or a different member of the same family? that the immune system is kind of locked in and might not be able to adapt properly to that. And you can actually end up with a worse infection than if you're never vaccinated in the first place. It's something that you have to be aware of. Both of these, in theory, they can be made fairly quickly. Again, the biggest challenge is probably going to be the scale. What's nice about these is, if you have the full length of a protein, when it gets made in the human cell, it's going to be properly folded, right? And then if it's in a human host cell, it's also going to have the proper post-translational modification pattern or glycosylation. Some of the caveats, DNA, right? There's a potential to integrate into the human genome, right? That's something that's really, it's out there. It's never been fully addressed. Maybe with some of these recombination systems where it can be that we're developing through gene therapy, that it can be trained to integrate into regions of the genome where it won't cause harm. I think that will be the biggest advance to come in the field, but it isn't quite there yet. I mean, they talk about CRISPR technology. In the clinic, they haven't had the best immunogenicity. And then for RNA, right, they're big time concerns with instability. And again, this is in humans and in a lower immunogenicity. Some of the other studies that are peer-reviewed that I talked about in the earlier bullet points, those are in animal systems, and it gets a little bit different. Once you go from a mouse to a human, it can be quite a bit different. Now let's talk about scalability, just to give you an idea for how much can you really make of these RNAs or DNAs. A lot in the news about that. Wanted to investigate it a little bit. I went to the press release from Nito Denko of Visha. Right, they're talking about being able to make to make DNA at a 1.6 molar scale, which is it's pretty good to get, and that's the largest. You can go to that web link and see. So in theory, if you had a 50 mR and you wanted to make a 100 microgram dose, that would translate to 264 million doses. But that's a big theoretical leap. That means just because it's made, you still got to purify it. Multiple steps in the purification, so I would think you wouldn't even get close to that yield, and that's still just a very, very small molecule. So I'm trying to give you a feel for the challenges that are out there to make something and have something that's widely available to many people. So I'll continue on a little bit more about RNA vaccines, some more bullet points, what's going on there. So in addition to something that's being either it's chemically synthesized or it's biochemically synthesized, you have a nice little feature here where it can be self-amplifying, right? So it contains the genes of the RNA replication machinery, right? Where the structural proteins, they replicate along with the gene of interest. So this may be difficult to do at the GMP level. There's some great papers that are there in peer-reviewed publications, studies where they show that it kind of almost mimics an infection in a way. So I think the closer you are to mimicking the infection, using the immune system's, exploiting the full capabilities of the immune system, the better you're going to be. So this is something that bears watching, and it could be the way of the future. Right now, I think it's something that probably couldn't be done so easily at the GMP level, but time will see. But with all RNAs, you've got some issues with stability, right? You can't really make something, have it distributed, and have it unstable, right? It's not compatible with developing a successful vaccine. You have to have some sort of shelf life, and if it's just something for just like months, it's not going to cut it. So you've got, these are some reports that are out there. So, freeze-dried mRNA with trehalose or naked mRNA, it's stable for about 10 months at 4 degrees Celsius. That's not great, but at least it's something. If you encapsulate mRNA with cationic liposomes or cell-penetrating peptides, right, you can they can protect it from RNAs. But I don't know what happens if you have some sort of excursion in the temperature, how good that's going to be. And then there's another all reviewed in the article below, so you can read a little bit more about it. A lyophilized mRNA vaccine was stable for 5 to 25 degrees for 36 months, and 4 degrees for 6 months. So you see that's getting a little bit better, and something that's more usable in a commercial context. So it's really would appear that you need to either have the RNA or the DNA either encapsulated or lyophilized, right? Just to use it naked or in solution, it will just never hold up. All right. So if you ever try to, whenever people talk about if any oligonucleotide or even peptide also is made synthetically, right, you've got to look at the link, the link between length, coupling efficiency, and yield, right? So it's a series, if you make a 50-mer, you've really got 50 different reactions going, right? 50 sequential reactions linking one base to the next, right? And the higher that efficiency of coupling, the better yield you're going to get, right? And you can see, if you look at the coupling efficiency, just a few percent, right? It's a very good rec, 97%. I mean, just when you start getting a much larger oligonucleotide, it really drops precipitously. So that's something to keep in mind. And there's no straight coupling efficiency across the board. Each oligonucleotide or each peptide, they're different. They're studies in and of themselves. So that's another challenge for manufacturing. All right, virus-like particles, you hear a lot about this. These are commercially available. They have been licensed. So, it's a good technology. It works, right? Again, sub-assemblies of viral structural proteins. Really, it's great. They're non-infectious, right? They don't contain any viral genetic material. They can have a very high antigen load. They resemble the parent virus taken up by cells, right? And they're processed by your immune system very similarly to the way that the parent virus from which they were derived is processed. Like I said, they're employed in licensed vaccines. They can generate cell-mediated and humoral immunity. The VLP, right, you can bulk express it in a bioreactor culture, right? Again, like I said, you do anything you want to commercialize, you can't do that unless you can really make it. And make it at large scale, right? Again, there's, I'll talk about the challenges also in case you want, you're looking at a different technology, you want to understand it, or a different company, what they're up to. There are some challenges again with stability, right? So you're challenged a little bit like that. For example, right? Commercial HPV vaccine Gardasil, right? Has to be refrigerated at 2 to 8 degrees, protected from light, and you can't freeze it. There's also, analytical challenges in their characterization. If they get stressed, that can change the particle size distribution. Also can affect stability. So they're kind of a little bit on the fragile side, but really not anywhere near what you'd have to worry about with an RNA. All right. Now we're getting more to like the live vaccines, right? So this is an actual, a virus that's live, can cause an infection, right? So, most of the viruses out there, that most viral disease, right, is or immunity to most viral diseases is best achieved by live attenuated viruses. Just think about historically what we've seen. We've seen like polio, right? Some of the first early polio vaccines, the smallpox vaccines. A lot of viral infections, right? The pathogenesis is complex, right? It's dependent on amplification of more than one target organ, right? You think of like viruses such as measles and yellow fever viruses. They don't just replicate in like a muscle cell or by shot. It's something that they need. Their natural life course across the context of the entire organism. So again, there's a concern, if ever you use a live viral vaccine that's attenuated, there always is the possibility of reversion to virulence. That was a concern with the early live polio vaccines. Now we're kind of moving into this replication deficient virus mode, where those are much less likely to have that happen, right? Since they by design they don't replicate. So that's a very good approach where they infect the cell, they just go through and it's just one round of infection. So you're getting the best of both worlds, right? You're getting an exposure to a viral infection, you're mounting the proper immune response, but you're not risk getting a full-blown infection, and you don't risk reversion, right? And then these also interact with the innate immune system. I mentioned that before. right? They're recognizing potential pathogens by the different molecular pattern recognition pathway, right? And then data from animal studies has been pretty consistent. They've indicated that not only are these live experimental, in the case of flu, right, most effective, but the directed, if you administer them via the actual way that people typically get infected, they yield better protection than generated by parenterally administered and activated vaccines. So I was just going to show you a little bit. Whenever you develop a vaccine, you've got to balance immunogenicity versus reactogenicity, right? And I couldn't find one from an actual study that was actually an actual virus that was administered, but I did find one that was actually a peptide vaccine. And I only include this because it shows you what can happen if you get a real sort of rough reaction. So anything that gets to the clinic, really, you can't have a lot of, swelling, soreness, fevers, fatigue. Kind of interesting, if you have a pet and you look and see after it gets vaccinated, typically they're not doing so well in the next day or two. But you'll never see that for an actually approved human vaccine. And I guess, Samuel, it takes us to our next question. I believe I had a question about would you be willing to tolerate a very reactogenic vaccine, or how many people would be willing to take a vaccine that's very reactogenic, but at the same time it's effective? I guess I wanted you to feel is how much are folks willing to be subjected to? Would you be willing to tolerate like having like a golf ball size welt if you knew you're going to get protection? Or would you just alternatively say, gee, that's something I'd really rather forego and just kind of take my chances because it's just not worth the pain and discomfort? Yeah, so most people would not want the golf ball size welt. So you can see, that people aren't, are much more discriminating than their pets. They would never accept something like this. All right. So again, recombinant vaccines, right? So again, these are things that you can make in some sort of incubator, right? I'll talk a little bit about the different cells that are used. Some folks have asked me in the past, what's the most common cell type? There are a zillion one of the, there are tons and tons of different cells that are out there, cell lines. The CHO cells, Chinese hamster ovary cells, they're sort of the workhorse for the therapeutic proteins. And about 70% of today's therapeutic proteins are made in CHO cells, right? And I put the reference there so you can, you can look this up and see what information you glean from it. So, the major step, right, the upstream processing, you gotta have a frozen vial, or you're gonna have to master it. That comes from a master cell bank. And these are common paradigms across all sorts of like cell culture-based vaccines. You get a producer clone, right, and it ends with the harvest of the culture broth, right? And then, here's some of you've got to have critical parameters. You don't just put it in there and it just magically grows. You've got to get the right cell density. You have to have the duration of a production phase where you get the maximum amount of accumulation of recombinant protein. You got to find out what's the best density, culture conditions. You got to have a defined culture media. You got to find how long the process has to go for. Probably I'll just switch back to this one. Yeah, just again for the different cells here. This just shows like the past 10 years and total and all the research. So you can see there are different cells, the HEC cells, the BHK, NSO. But you see what's consistent over the, looking at, again, here's the reference where it came from. Over the course of the last 10 years and total things in PubMed, that the percentage of CHO cells, it's pretty constant. So that's kind of like your workhorse, and that's something that that's sort of the most common cell type. And then just to give you an idea, of how big these things can get, you can have up to like a 20 kiloliter bioreactor. I know a lot of folks have sort of asked me how much can you make and what's the size, and then you can get but these take for a while to do. It's like 29 days and you get 2 tons of cells, up to 2 tons of cells to be generated. So that's pretty good. And then for your yields and stuff, if you think about dosing and getting something to many people, you get production titers, close to anywhere from 1 to 10 grams per liter in the different batches, depending whether it's like sort of a batch mode or a fed batch mode. You're continuously replenishing reagents. And, you know, whenever you're working with like a sort of a recombinant protein like that, you've got to try to take measures to mitigate against the loss of biological activity, something that might diminish or it might cause unwanted immunological reactions or other side effects. So you're going to have to do a lot of steps for quality by design and analyzing protein quality. All right, this takes me to my next slide, my next little pathway here. You know, maybe how to think about these things. And again, I'm trying to present something is what's the fastest, but at all times not by no means compromising safety to kind of get a vaccine that's useful, and maybe how we can maybe trim the 10 years down as best we can without doing something that's completely reckless, right? You know, and I put a little flow diagram together. You know, so people can kind of ask themselves these questions. You first, you know, if a virus can be grown in cell culture, right? You know, then that's great. You know, then you might be able to do multiple passages or figure out a way to get an attenuated virus, and that's probably a way of getting a lot of virus and a lot of vaccine. You know, if it's reactogenic, then you'll have to think about like somehow inactivating or splitting it. If not, then you can certainly go the routes of VLPs, right? You know, and then you kind of sort of work your way down. If no, right, then you know you're going to ask, you know, if maybe just neutralizing antibodies would just by themselves would suffice. Then maybe you can get by with a recombinant protein. And I've sort of put a question mark here for the oligonucleotide. That's not, you know, since there's really nothing that's been commercially licensed, it's still really not clear exactly where it sits along the scheme. Then there's some other technologies that really are not on the slide that I just really didn't mention. There's been different techniques or different ways that you know, in aquaculture and agriculture, some very successful ways to develop vaccines, and that might be something that we should emulate in people as well. You know, there are different vaccines for fish to get different diseases. There's where they take that orally. There's even a coronavirus. I'm sorry, yeah, the coronavirus type vaccine in chickens, believe it or not, and that's for an infectious bronchitis. That's a coronavirus, and that's actually been tested to work and protect chickens where it gives it to them orally. So these are things that maybe at some point they'll find their way into this flow diagram as well, or should be thought about. All right. So one really important thing when developing a vaccine is to have a correlative protection, right? You've got to try to figure out early on if you're generating an immune response that's actually going to translate into protecting a person against the disease when they're exposed to the live wild type virus, right? So you got to be careful because some of these animals really don't mimic humans very well. A lot of stuff's done in mice, but the mouse immune system is quite a bit different. In fact, I give you a reference here. You can look that up. Don't take me for my word for it. You can see for yourself. And if one's going to use a mouse, it's probably better to use one that's actually humanized, where you have a human immunological synapse, something that would better recapitulate disease. But you have to be wary of these mouse models. Obviously, a mouse is the cheapest system available, a cheapest mammalian system, so that it tends to be used quite a bit. But other things are much better. Like for flu, probably a ferret is a better animal system to work with. Certainly, primates as well might are that much closer to people. So you got to be aware of some of these things. You have to have proper potency assays, right? They can be cell-based, mechanism of action, immunochemical. You really want an immune response that's responsible for and statistically interrelated with protection, right? It makes no sense if you get a great immune response, but it's really not going to protect you. I mean, you'll get maybe something in early clinical stages, but you really won't get something that goes much further. That's something to be aware of. All right. sorry, you gotta have, now we're talking about like actually manufacturing something. So, you know, it's one thing to have an idea, right? It's completely different to have a manufacturing process. One thing I never really appreciated when I was in academia is just how difficult it is to have a good manufacturing process, right? It's a science in and of itself, right? The initial identification of the epitope, the response you get in a preclinical animal system, those are all very, very upstream, right? But you still really haven't answered the real crack the tough nut. Like, how are you going to make this, right? I mean, there's a million things that are going on, right? And how are you going to make it reproducibly? You can't just make something that works, some of the time or once in a while. It's got to work every time, and it's got to have the same level of potency each time. Otherwise, you can't, you'll just be rolling the dice with each lot. So one thing that's real critical here, right? Something called the design of experiments, right? DOE, right? So a lot of times, if you don't have a DOE, where you look at different factors in your formulation, in your process, you'll just be doing things by trial and error, right? I would say anything you get involved with, any process, make sure somebody's done a DOE experiment, right? There's a great paper here or chapter in a book, right? You can look it up, right? You can save up to 50% of time by performing a proper DOE. And whatever you do in terms of your formulation, the DOE should also kind of look how it interacts with a process, right? You don't do those independently, right? You got to develop your process as you're getting your formulation, right? You wouldn't want to do some sort of initial study, have great potency in mice, and then when you finally make it, you don't really have, you find it's really not nearly as potent as it once was. So that's something very much to be cognizant of, right? Another thing is, like, if you're trying to go for a live vaccine, right? I talked a little about this in our flow diagram. Some of these viruses just can't be grown in cell culture, right? That would be something like HBV, right? Human noroviruses, or they might have might not really produce yields of progeny that are high enough for use in a vaccine, right? HIV, right? HCV, right? Other herpes viruses, right? HIV, HCV, those are really kind of been notorious for we've worked many decades on those, and we still have no vaccine. So it's like I said, it's definitely a worthy endeavor, but it's something that really it can be very challenging. And anything you make, you've got to also, in the manufacturing strategy, you've got to have mechanism intact to minimize any post-translational modifications, right? You don't want microheterogeneity, right? You don't want to, they could have an impact, right, on solubility, potency, biological activity, all these things you have to take in mind. And then when you actually make it, you probably, in terms of speed, you're probably not going to, you want to stay away from something like a traditional stainless steel tank. Single-use bioreactors are the way to go if you're making your product in a bioreactor, right? You don't have to validate those. They're reliable, right? They're clean, pre-sterilized. So you don't have to deal with all the requirements for installation, qualification, and personnel training. So those are just some points to keep in mind. All right. So another thing to kind of speed stuff up, right? So the critical path, right, for IND filing, right? It's the availability of your tox lot for toxicological studies. So you want to get that going as soon as possible, right? It takes about 6 months. So you don't want to have that at the very end, right? Because they'll just add like another 6 months. It can't be, it's got to be done in parallel, really not in sequence, or not sequentially. You could do it like that, but it's going to take longer. Your investors are going to get mad. You might run out of money. All these are steps to kind of trim the path. You're not cutting any corners. You're just doing things efficiently, right? For process development, you want to get that done as early as possible. Your formulation development, method development, those are all concurrent, right? Again, you're doing these in parallel, right? If you're making clones, you get these things done before screening the actual clone. Sometimes for your tox lot, right, you can, if you're working with different clones, you can use a pool of clones, right, that will be acceptable before you get the highest yield clone. So this way you can get your stuff in tox as soon as possible, right? And then you want to have your GMP production start, you know, prior to the full completion of the toxicological study. You can kind of get a little bit of a jump start, right? And as I said, the toxicological study can be on a pool of material, right? So you don't have to necessarily wait till your very best clone comes available. So, all right, I talked about manufacturing, right? Some of the strategies there, right? What are some of the other levers we can pull? So there's certainly regulatory mechanisms. Now there are four, I list here 4 different pathways or 4 different mechanisms. They're really 6, right? And I'll talk about this a little bit because you're hearing a lot in the news, and I'd be remiss if I didn't mention those, right? So just a couple things, right? When they talk about approval, right? Or designation, they usually accompany these terms, right? The approval mechanism, right? Approval is just simply a mechanism to market authorization, right? And the designation, right? It's granted to a drug based on meeting certain criteria, providing certain benefits. So that's a distinction between approval and designation. And again, here are the 4 main expedited pathways. But there are a couple other ones I didn't quite talk about, right? There's another one called emergency use authorization. You hear a lot about that, and that kind of scares me. Because there, I think you might be taking away some of your safety mechanisms. Let's say I wanted to drive from New York to Boston, right? And I wanted to get there faster. Well, there are ways I can do it. Well, let's just say, you know, I could go through stop lines, stoplights, stop signs, take the brakes off my car. That would get me there, but you know, it would be a perilous ride. And basically, the fast track designation, it gives the FDA authority, right, to the regulations on that now, where they have the authority to actually, where they allow an unapproved medical product, right, or unapproved use, or the unapproved uses of approved medical products, right, they can be used in emergency to diagnose, treat, prevent serious or life-threatening diseases or conditions, right? And this is generally for CBRNs, right? Chemical, biological, radiological, nuclear. So yeah, so that's that. So it's like for the CBRNs. So that's kind of what it's used for. What scares me about it is something could be, could get into use much faster than it should be. I don't see it as a real typical thing. I talk about it because of the current times in which we live. In that, in conjunction, there's another program that's out there, or I should say there's a Supreme Court case many years ago. Actually, it was called Jacobson versus Massachusetts. No connection to me. It was in 1905, way before my time. Suppose with a name like Jacobson, he was a great guy. but in all seriousness, that basically gives the state the authority to give you a vaccination at their discretion. That was done in response to smallpox. So that's a fast way of getting things into use. But I don't think it's it's something that's certainly not a normal development pathway. So I'll say a few words about this. We've got the accelerated approval, right? So that's the accelerated approval on number 3, right? That lets drugs for serious conditions, right, that fulfill an unmet medical need, right, to be approved based on a surrogate endpoint, right? I'll say a few words about priority review, right? The designation means the FDA goal is to take action on an application within 6 months. So usually it's about 10 months. So that's a way to kind of, if you get that fast track designation, right, you're going to get a priority review. You're going to get your drug, your vaccine developed much faster. Fast tracked, right? That also develops the gets a faster. It's also for an unmet need that gets developed very quickly. And then you've got the breakthrough therapy designation. So those are all the 4 mechanisms. The FDA is very involved in those. You've got to request those pretty early. And then I'll say just a few words before I move on. I see we're getting a little close on time. That you also have a voucher program, right? For tropical diseases, neglected diseases, orphan diseases that affect less than 200,000 people. If you're an entrepreneur and you get one of those vouchers, right? Your product, you request that during development, you're getting a priority review for any other product that you want to develop in the future. So the beauty of that is that you can actually sell these. Well, if you've got a product, it can be developed faster, but you can also transfer that to another entity, and typically those have a pretty high market value. We're talking like, $200 to $300 million. So if you've got a small company, that's certainly a strategy to consider, getting some sort of voucher, priority review voucher that can be sold. That would certainly, be quite a boon to your financial resources. So I'm gonna go through the rest pretty quickly. You guys can look at that and see we're getting close on the hour there. Stability. There's a big, is a big commitment that has to be met, right? I've put up here. Here's the stability for phase 1, the requirements for phase 2, for phase 3, everything that's expected. So I just wanted to give the bio entrepreneurs in the audience, and also folks that have an interest in vaccines, what's really required to make a good vaccine with regard to stability, right? And then I'll finally conclude with a few words on opportunity. So irrespective of what you think about the fellow who originated this quote, when you sort of read this, as we know, there are no knowns, there are things we know. We also know that there are known unknowns, and that is to say, we know there are some things that we do not know, but there are also unknown unknowns, and those are the ones we don't know. So kind of painful to read this, but after about the 10th time, you realize there's a certain amount of wisdom there. So what do we got here? We're concluding on the opportunities. We've got the known knowns, right? Those would be viruses that just are re-emerging, right? Known unknowns, things that are emerging, right? And again, I'm gonna say right here, like, most of these actually are zoonotic. They come from animal species. So it's something that's a pattern that's gonna probably reproduce itself. I would see our opportunities are in the known knowns, the known unknowns, right? Unknown in terms of how infectious they're going to be, and then something that's completely unknown, we really can't do much about. And really, I'm gonna, after the final few slides, I'm just going to devote to like the viruses that I call these like the known unknowns, the ones that really don't have a vaccine. We know about them, but they don't have a vaccine. And that's sort of a potential opportunity. I put the little iceberg here because in terms of tip of the iceberg, I mean, it's not even what I will present towards the end. It's really just not even a snowflake on the iceberg. I put a quote up here from a book from The Coming Plague, right? And in a nutshell, what it's saying is the more the world develops, the more we interact with different folks, the more we're putting ourselves at risk to encounter one of these zoonotic organisms. So even though this was written in 1994, I actually got this book coincidentally in December of 2019. I thought it would be interesting reading. I never knew it would be so appropriate. But you see, this is going to be a recurrent paradigm, right? I know there's talk about the current situation that it's a bioweapon or escape from a lab. I kind of hope that I wish that it were, because that would mean it's a one and done issue and it could be addressed. But if it's most likely if it's something that came from nature, you can see that this is just something that's going to happen over and over again. And the situation we find ourselves in, it's really a light tap as opposed to what could be a significant punch. If you look at these, we'll conclude by, I'll show you the viruses that are out there that are completely, that we don't have a licensed vaccine for. It's a scary list when you look at some of these, when you look at the mortality rate is, when you look at the symptoms. If these ever became like, widely infectious, it would be very, very problematic, much worse than the current situation that we're now in. You just go down the list here. I've referenced the different studies on the ClinGov trial site, so you can kind of get an idea what's out there, and where we stand. But it's sort of a frightening list, and what kind of terrifies me is some of like the cutest organisms really are the most problematic. And I put up here a squirrel monkey and a spider monkey. These guys harbor viruses like you wouldn't believe. You'll never go to a zoo and you'll never see an old world monkey mixed with a new world monkey. That's been tried, but these guys have so many viruses which they're immune to, or what they really have. These herpes viruses, they actually they spread very readily to the old world monkeys, and they give them like a cancer or leukemia where most of them end up dying within several months. So heaven forbid that something that, due to gene swapping could ever find its way, and it potentially is possible, into the human population. So this kind of gives you a glimpse. You got this long list here. Here's some like known unknowns or known knowns that we can, maybe it's worth going for one of these vouchers or orphan diseases and developing something against this. So, having something in our arsenal if the unthinkable actually happens. And with that, I'm going to get my concluding slide and see I'm close to time here. And I don't mean this disparagingly in any way to anybody's efforts. Just all I mean to say is that it's a very iterative process, vaccine development, right? The first vaccine is probably not going to be your best. It's just really the first horse out of the starting gate. And I borrowed this quote from a gentleman who was was a very well-respected and well-established gene therapy researcher, which I heard him say at a bio conference. Just because a horse is first out of the gate does not mean that it's Secretariat.